So this is what we've got to do for the rest of the semester. So we're behind. We're going to kind of keep these dates. So I'm going to tweak it a little bit. So exam 3, it's supposed to be online. We're doing it on campus. So we'll do that Wednesday. I'm not going to get into this material today. So I'm like a week behind. So I want to do exam 3 on photosynthesis because that's what we took our quiz on. So I'll put some of those questions and I'm going to go over it again today. And then I'll give you some questions and then I'll introduce plants, which hopefully is a nice and easy chapter, I hope. So I'd like exam 3 to be Wednesday in class on photosynthesis in plants. So really you'll just have one small chapter to study. Then quiz 4, we're gonna keep that on schedule and put that online, and it's only going to be on one chapter. So that'll be just the next chapter, animal tissues. So exam 3 is on these 2 chapters, photosynthesis and plant structure. Quiz 4 is just on animal tissues. That'll be online, just like it says up there, from October 2nd to October 6th. Then, and so I've got a video on animal tissues. I will post that. I'm going to wait until Wednesday to post it because if I post it now, some people are going to study it for the exam. And but if you want me to like email you a link or something, I can. It's on YouTube. If you just put my name into YouTube, you'll find it. Like you look for like the folder for biology, and then you can see the video. So it's not like you can't access it now. And then cardiovascular or animal nutrition and cardiovascular. I'm gonna lecture during the first week in October. So it says here October 5th, review in class. I'll probably do a class. So I'll probably extend it a little bit. So, so this week, today, go over photosynthesis a little bit and go over plants. Wednesday, take our exam in class. So there won't be really a lecture. I'm going to put a video up for animal tissues. So that's going to be quiz 4. Then you take your quiz like over the weekend between Friday and Tuesday. And then the next week, so the week after this week, I don't even know what the date is. The 28th? So how many weeks is actually free? Hold on, let me just. So we got this week, and this weekend's October 2nd. Yeah, so next Friday, is that what it is? And I know it's a Friday, but that's like what it is on the schedule. So I've got to keep it. I've got to keep the exam 4. They give us certain days, and I have to keep it on those days. So next Friday is the end of class. So today, photosynthesis in plants. Wednesday, take an exam. Instead of me lecturing Wednesday, I made a video. Next week, we're going to meet Monday, and I will go over digestive. On Wednesday, I will go over like the heart, cardiovascular. Right? I'm not going to be able to do the whole chapter, right? But I'll go over. So that's my plan. So next Monday will be digestive system. Next Wednesday will be the heart. And then next Friday is the final on campus. We'll use the same times that I've been using. I'm going to make an announcement with all this, but I just wanted to put it out there. So the only thing left for you to take this online is quiz 4. Exam 3 and exam 4 are both on campus. I'm going to change the nature of the questions for the exams. I'm going to make them like essay, short answer essay type of things, because grades got a lot better with those types of tests. So I'm not doing it as a punishment, I'm doing it to make the grades better. So that's the plan. And again, I'll send out an announcement about that. Look, there's the problem right there. I've got 4 viewing plans out of like 30. It's fine for me. All right, let's go over. Let's do. I got two chapters. What do you want to do first? PowerPoints or plants? Photosynthesis or plants? Do you even care? I'm going to do plants first then, because I have the video already up on photosynthesis, but I don't have one on plants. So I'm going to do this one first, in case it takes me longer than I want. You guys seeing the board right now?
Yes.
What do you want me to do? All right. So hopefully an easier chapter. So I have a PowerPoint that, I'm gonna present this off the PowerPoint, but I'm just gonna write for a second as well. So plants, they're different in a couple of ways. One, plant cells have a cell wall. They have chloroplast, which I can't spell. That's the part, that's where photosynthesis happens. And then we need a third thing. They have large vacuoles. A vacuole just holds fluid. Maybe not. Is that blurry for you guys?
It's a little blurry, but I can read it.
Let me see if I can do something about that. I can't. All right, well, okay. Don't worry about if it's blurry, because I'm going to, no, it's, I can see it, it's looking a little blurry from my end. So, I'll try to write a little bit bigger. I'm only gonna write a couple more words, and then I'll just try to do it off the PowerPoint. So, we've divided plants into two types. Plants that have flowers and plants that don't have flowers. So the plants that don't have flowers, none of these plants work, are called gymnosperms. So gymnosperms don't have flowers. So like, for example, pine trees, mosses, ferns, right, they don't have flowers. Those types of plants probably came first, right? And so the plants that we're gonna spend time talking about are called angiosperms. So those are flowering plants. That's it. No flowers, flowers. So I'm gonna, since it's kind of blurry, I'm gonna switch to it still looks weird. I'm gonna switch to PowerPoint, and I'm just gonna go off the PowerPoint. You guys see my PowerPoint?
Yes.
Awesome. All right. So within angiosperms, what you see up in, I don't know why I'm standing in front of it. What you see up in front of you are a way to delineate two types of angiosperms. So one type is called a monocot, and one type is called a dicot. The monocots and dicots. The cot part stands for cotyledon. So if you look at like this word here, you know, cotyledon is like the first little leaf that comes out. It's not really a leaf, but it's like the first leaf-looking thing that comes out of a seed.
Professor, sorry to interrupt. It's still showing you in not presentation mode. It's just showing all the slides planned out.
Oh, it is?
Yeah.
All right. What about now?
Now it's opened up again.
All right, so you see just that one slide?
Yes, with monocots on the top and dicots on the bottom.
Yeah. So the cotyledon is just like that initial leaf that comes out, and it's not like a real leaf because after a few days or a week, whatever, that little leaf will die, and then you'll see the actual leaves come out. But it's just like that little green leafy thing that pokes out of the seed just to get the plant going. So monocots have only one of those coming out, and dicots have two of those leaves coming out. So that's one of the differences. And then in monocots, and I think of like, you know, like the top one you can imagine is like grass, and the bottom one would be like a tree, like an oak tree or something, right? So if you look at the leaves, the veins are parallel. So the veins in the leaves carry, and we'll talk about them in a minute, leaves carry water and sugar in them. So in grass, you'll notice that they're parallel. Or like another good example, I guess, would be celery. Like it's all stringy, like all those leaves are all parallel. Whereas, in the dicots, they're not, they're more like nets. Vascular bundle, we haven't gotten to this yet. So, just look at it for now. With monocots, they're not arranged in any particular order. So if you look at again, celery, it's all, they're all parallel, but they're all in there and there's not a particular arrangement of them. Whereas the stems in dicots, they're more orderly. And you can kind of see it right here. All right, but let's just get past that. Let's look at the roots. So again, if you think of grass, you can pull up grass and plant it somewhere else. The roots are very close to the surface. They don't go down very deep. Whereas trees will usually send down a taproot and have some lateral roots coming off of it, right? So dicots send their roots deep to get water. Monocots, like grass, keep their roots up towards the surface to get water there. So fibrous, or sometimes they'll call it an adventitious, fibrous root system. Fibrous root system versus taproot system. And then last, the flowers are, and we never let grass go all the way, but the flowers are in multiples of 3, or in dicots are in multiples of 4 or 5. So those are kind of like differences. you know, 3 parts to a plant, pretty easy: root, leaf, stem. There's no need to memorize all these parts. It's just kind of showing you, you know, there's a taproot, and there's the lateral roots. I'm not going to ask you a question about that. I'm not going to ask you about the petiole, the blade. You know, I'm just kind of showing you what it looks like. There is something I'd like to show you, the term apex. The apex is the very end of something. So in you, the apex is like your toes and the top of your head. Right? So the apex is the very end of something. And we use that word in anatomy as well. So you have an apex bud, if you see at the top, that's going to grow the plant taller. You also have the apex of the root, the tip of the root. So the plant grows, you know, deeper or taller. But then you see axillary buds. So if you look at axillary, that's kind of like the armpit of the plant, right? So that's what we call our armpit section. So axillary and apex, those are 2 anatomy terms as well. Right? That's just like a little FYI thing. Okay, so cells. There's 3 types of cells in a plant. So I definitely want you to know this. This is something that I'm going to ask you on the test. Like I said, I'm coming straight on, I'm telling you. Most likely. I might forget when I'm making it, but it's something that I almost always ask. 3 types of cells. So you see A, B, and C. Both of these are the same at the bottom. So let's go to A first. Parenchyma. So when you see often in anatomy, when you see a word that has a CH followed by a vowel, like CHI or CHY, it's pronounced with like a hard K. That's just like a little, for your information type of thing. So parenchyma cells, the difference between all these cells has to do with the cell wall. So remember, plant cells are different than our cells in that our cells have a phospholipid bilayer. That's like our membrane, right? Plant cells also have that, just like we do, but they also have a cell wall around that. So I always make the analogy like our cells are like balloons. Plant cells are like balloons inside of boxes. So the balloon will never pop because the box prevents the balloon from getting too large. Right, so the box in this analogy would be like the plant cell wall. So boxes are like thicker and thinner, right? There's like a double-walled box, single-walled boxes. That's kind of like this. So parenchyma cells have thin cell walls. So I can't write this part down. You're gonna have to like make a note. They have thin cell walls, and they're like generalist type of cells, meaning that you find them in lots of places. So you find parenchyma cells, for example, in leaves. The cells in leaves that go through photosynthesis, they're parenchyma cells. The cells in roots are like a potato. The potato is just not technically a root, but anyway, potatoes are like parenchyma cells. Right? So you find them lots of places. So two things about parenchyma cells: thin, flexible cell walls. Number two, they're like general cells. They're not specialized. Parenchyma are specialized. So we're on the second type of cell. Parenchyma cells you find in new plant growth. So here's the key term. Parenchyma cells have unevenly thick cell walls. So parenchyma are thin, parenchyma unevenly thick. Some parts of the wall thick, some parts of the walls thin. Because you want to provide support for the plant, but also the plant has to grow. So you find this with like new plant growth, like new shoots, plant shoots. The last type of plant cell is called sclerenchyma, and both of these at the bottom are sclerenchyma. And I'm not gonna, you don't have to know the two types. It doesn't matter to me. Just know sclerenchyma cells have a very thick cell wall. So I'll give you an example of sclerenchyma. Anytime you're crunching some kind of fruit, you're crunching sclerenchyma cells. That word S-C-L-E-R means like hard. Like it's in the word, for example, sclerosis, scleritize, it means to harden. So sclerenchyma cells have like hard cell walls. Wood, for example, wood is just a bunch of sclerenchyma cells compressed. I'll give you another example, like the like seeds, like the shell of seeds. That will be like sclerenchyma cells. So they can be quite hard. So that's it. That's the three types of plant cells: parenchyma, parenchyma, sclerenchyma. It just depends on the cell wall. Parenchyma thin, parenchyma uneven cell walls, uneven thickness. Sclerenchyma thick cell walls. This is an example of a sclerenchyma cell. So this is called, these are called xylem cells. Now you might think of cells as being round. They're often portrayed as being round. But this cell here, so if you look at this cell, it's really long. So this whole long thing is a cell. Actually, there's 3 cells here. Like this one here on the right is 1. And then there's 2 cells on the left here. So they're elongated cells. This is partly what makes up like the vein of a plant. So there's actually 2 vessels. There's 2 types of cells that make up veins of plants. One is xylem. Xylem carries water. It's that simple. So xylem carries water. These are actually hollowed out cells. So they're kind of like straws, right? So it's a hollowed out cell. It's dead. It's actually a dead cell. It's a dead cell, hollowed out. There's nothing in it. It's just the cell wall. But this xylem, that's what wood is. Wood is just xylem. And so these cells, these are a type of sclerenchyma cells. They're rather hard. I mean, even if you take like a leaf of a tree, right? There's some softer parts of the leaf and there's some harder parts of the leaf, right? So the veins of the leaf are much harder. Like you could maybe snap those, but you couldn't do that with the other parts of the leaf, right? Because the rest of the leaf is parenchyma. The veins of the leaf are sclerenchyma. So you can kind of see the difference. The other part of the vein is called phloem. So it's up at the top there, P-H-L-O-E-M. Phloem carries sugar. So you got these. Let me let this person in. You got these 2 vessels. One's carrying water. One's carrying sugar. So one's carrying sugar, one's carrying water. Phloem carries sugar. I'm not going to ask you about all the parts or anything. I just want you to know phloem carries sugar. And it's another example of sclerenchyma cells. These cells are actually alive. They're living, but they're always next to each other. The xylem and the phloem are always next to each other. So when you're looking at the vein of a plant, you don't see it, but it's two different cells together. Xylem and phloem. So you've got a separate vessel to carry sugar and a separate vessel to carry water. Any questions so far? I'm recording this, by the way. Okay, so what I gave you was like the xylem and the phloem. That was a type of sclerenchyma cell, but it's also, we call this vascular tissue. So plants have 3 types of tissues. So first of all, we should say what a tissue is. Tissue is a group of cells that have a common function. For example, your skin is a certain type of tissue that's different from your muscle. Your muscle is another type of tissue. They're all cells that come together for like a common purpose. Right? So in plants have 3 types of tissues. We have 4. We're going to do us next lecture. Or actually that video. Right? But right now we're on plants. Plants have 3. And it's just what you see in front of you, vascular dermal ground. And we can figure this out, well, we can figure out two of them already. Dermal, you're thinking skin. So yes, that's dermal tissue, it's the outer layer. So if you think of a potato, the potato skin is the dermal tissue. Or carrot, a carrot's better. All right, so let's think about a carrot. You skin a carrot. I never do, I don't know why you guys bother. But you skin a carrot, that's the dermal tissue. Now when you look at the carrot, you see that cylinder in the middle, right? That's, in that case, that's the vascular tissue. It's right in the center of the carrot. The rest of it is called ground. So this is how I define ground tissue. Whatever's not vascular, whatever's not dermal, whatever's not vascular, whatever's not dermal, that's ground. So take the part of a potato that you eat, like the white part that you make French fries out of. All right, you skinned it, so is it dermal tissue? No. Do you see like any veins in it? I don't. So I'm gonna say no for that. So, then it's the part of the potato that you eat is just all ground tissue. It's not vascular, it's not dermal. So it's just all ground. If you're eating just potato skins, then you're eating dermal tissue. So that's the three types of tissues. So the xylem and the phloem, that's both together they make vascular tissue. In fact, the xylem and the phloem are always bundled together. So we call it a vascular bundle. That's the other word for like a vein. So we call veins like vascular bundles because they're made from xylem and phloem. You know what it is? It's like the wire to your cell phone charger, right? If you cut that open, you see that there's a red wire, a smaller red wire in there, and a smaller white wire, and it's bundled up, kind of like that. I don't know, man. My analogies suck. I don't know. I try. Here's a root. So let's look at a root and let's look at the different. Tissues, vascular dermal ground. So here's a root. You see in blue, that's the dermal tissue. It's like the skin of the root. And you'll notice here that there's root hairs. I'll show you another picture of it right now. Radishes have pretty good root hairs. So root hairs, you see these hairs, that's just part of the dermal tissue. That's just so it can get more water, right? So like if you look at the radish, it could reach out a little further and get a little bit more water than normal. And obviously we don't ever really see root hairs. They're hard to see. Radish has some pretty good ones. But anyway, that's the dermal tissue, the skin, right? And then the vascular tissue, so in roots, the xylem and the phloem have kind of come together. So if you go back to the carrot, you just see like that one cylinder thing in the middle. So we call that stele. So in roots, we call the vascular tissue stele. It's not xylem and phloem together. It kind of merges. Because mostly what's happening in the root is it's collecting water from the soil and taking it up to the rest of the plant. That's mostly what's going on. Not all of it, but mostly. And then the yellow is stele. Sorry, screwed that up. The yellow is ground, what we were thinking ground. So the ground tissue, it's not the skin, it's not the vascular tissue, so that's it, it's ground. This is, I'm probably not going to get into the meristem. I'm trying not to give you too much information on this chapter. Meristem is just where you have a lot of cell division, but we're going to skip it. So yeah, that's just showing you a root. I do want you to know that roots have root hairs. So this is kind of interesting. I'm not going to test you on this, but if you look at this onion, here's the roots. So what's this white part that we eat of an onion? If that's the root, then is that white part also a root, the part that we eat? You don't have to answer, I'm just thinking to yourself. It's obviously not, because I'm asking you. Right, so it's onions, think about them, they're leaves. They're different, they're kind of like special case leaves. But sometimes these plants aren't what we think they are. So this green part is the leaf, and that leaf extends down to the white part of the onion. And then the stem is like a little nub. Same thing like celery. We think that the part of the celery we eat is the stem. It's not, it's the leaf. The stem is the little nub at the bottom of the celery. Potatoes, we think that we're eating the roots of the potato. No, roots grow from the potato. From one end, the roots grow, and from the other end, you'll see leaves start to come out. So what you're eating is actually a stem. A potato's a stem, the part we eat. Anyway, that's none of that's on the test. I just, I have like this desire to say something that is interesting. Because I know what these lectures sound like. And I know that's not interesting, but whatever. It's more interesting than talking about root hairs. Okay. So root hairs. Okay, I'm just showing you. This is a slide from a root. And so you can see the dermal tissue is just right. It's kind of hard to see on this, but it's just like a layer of cells around. This one's easy. You can easily see the dermal tissue here. And look, the vascular tissue is right in the middle. So the vascular tissue is right in the middle. And when you look at the vascular tissue, you're seeing two types of things here on both of them. Like, look, here's the vascular. We call this the vascular bundle, right? Or in this case, because it's a root, we're calling it skin. But you see like those big holes? Like these big spaces here? That's carrying water. And I know like the smaller holes around it is carrying sugar. I'll show you in another. This is a stem. This should be a stem. Well, I don't see it. It's a stem. So these are stems, right? So in the dicots, you see that the vascular tissue is kind of like it's organized, right? So you're seeing here they've got it stained, so you can see colors, right? So blue, red, right? So the reddish color is the phloem, and the bluish color is the xylem. And the reason you can remember that, not that I'm going to ask you on the test, but the xylem is what makes wood on a tree. Phloem is what is going to make bark. And I'll talk about that in a minute. But the bark's always external and the wood's always internal. So the xylem's always internal to the phloem. This is on a monocot, so it looks a little bit less, or it's not organized really. You see, like I'm pointing the tip of the pointer, like the circles. It's not blue. That's all the xylem, and then the blue part is like the phloem. But as it's labeled here, it's bundled together, right? So vascular. Each of these are vascular bundles. And that's just, I'm just showing you what it looks like a slide. We never really got to do lab. So, normally I would have shown you these things in lab. We would have made our own slides of them, maybe. I'm going to skip this one. I was thinking I was going to give it to you in class. I decided to just not worry about it. That's just a leaf. You know what you can see since we're looking at it? Again, I'm not going to test you on this, but you see how there's just a bunch of squares? You see, when you look at it close, you see squares and dots, right? So these are squares all next to each other. That's the cell wall. See, they're packed in there. They're packed in there like bricks, right? You're looking at all the cell walls. When you're chewing on salad or something, that's what you're breaking these things up, right? So you make it fiber. All these cell walls you're breaking up with your teeth, and they're just the fibers, just the pieces of it. And then you see all the dots? All the dots are the nucleus. You can't really see anything else in these cells because, cells you have to give it a stain. You have to stain it. Otherwise, nothing's going to show up. It's really hard to see. So typically, we stain this stuff we got two colors, right? One's called eosin and one's called whatever, doesn't really matter, hematoxylin. They stain red and blue. That's why when you always look at these slides, you see either red, blue, or purple, or different shades of that, right? Because that's the stain that we use. It just depends on what you want to put on the slide. So, anyway, you'll learn about that in A&P, but you're seeing here a bunch of cell walls together, and all the little dots that you see, that's all the nuclei. That's what happens to stain when we stain these things. So all of the organelles inside, like the chloroplast or whatever, you can't really see them here. But here's a closer look at the leaf. So again, dermal tissue. This is kind of like a cross section of the leaf. Here's the top part, here's the bottom part. So you're seeing two dermal tissue. I'd like you to know this: plant leaves have usually cuticles. The leaves and often stems as well. They will have a cuticle, which is just a waxy layer. So often leaves and stems will have a waxy layer called a cuticle. And that's just to prevent desiccation, drying out. And look, here's, this is not to be confused with stroma. This is a stoma. See both of these cells? So a stoma is made from two cells. And this is like the pore of the leaf. This is like how the leaf will breathe. Let in carbon dioxide and let out oxygen. So again, the vein, in this case, the vein is pink, the vascular tissue, vascular bundle, right? Because you see here what xylem and phloem are together. So this pink part is the vascular bundle. The green in here, that's all ground. It's ground tissue, but what kind of cells are they? Well, it's labeled, you can see it, but it's ground tissue. You see all these green dots inside? Those are chloroplasts. So this is parenchyma cells. All these green cells in here, these are all parenchyma cells. This vascular tissue, sclerenchyma. They're calling this parenchyma because they want to put a new type of cell in there. So they're trying to show you that the leaf is growing. Like this particular area of the leaf is growing, so they just wanted to include it on the slide. Right? But just remember, parenchyma cells are wherever you have new growth. The key word, uneven. Unevenly thick. Sclerenchyma, very thick. Thicker, whatever. Parenchyma, thin. This is the last thing I'm going to show you for this chapter. And then I'll just sum up what it is that I want you to know from this PowerPoint. So you have two types of growth in a plant. A plant can grow taller, and that's primary growth. That's it. Plant gets taller. But some plants get thicker, like trees, right? They're woody plants. So they get wood on them, or they grow thicker. So I just wanted to show you how it happens. So what gets thicker on a plant is, so this is like a tree. So we're looking at the vascular bundle. So look right here with the part that's yellow and the part that's brown. The brown is the xylem, and the part that's in gold is the phloem. In between the xylem and the phloem, there is a layer that is going to divide every year. So I'm going to show you the next year. So this first layer, here's like after the first layer, this first layer right here got pushed in. I'm not saying it got pushed in, but a new layer came right here. See like this tan color? That's the new xylem. It's like really active. And this brown layer is not as much active. So there's lots of water flowing through this brown layer, but not so much water flowing through there's lots of water flowing through the tannish color area, not so much in the brown area. And then look on this other side. This is the new bark. It's also like tan. And the gold color is like the old bark. Or we could say the old phloem. Forget about the blue color for right now. I know they added the cork, but I'm going to forget about it for right now. So we're just looking at this. So right here where my pointer is, this gold color, that used to be the active phloem. There was sugars flowing through this. And now here it is. The newer phloem is right here. Now we look at the third year. So there's that first layer of xylem. There's the layer from the next year, and there's the newest layer. So, you get the idea. There's the oldest bark. Wow, where's the oldest bark? I don't know, I can't, it doesn't, I don't see 3. Oh, right there. All right. So there's the newest one. Next one. All right, so that's like the rings on the trees. Right, that's all it is. It's just like a new, as closer you get to the middle of the wood, we call that like heartwood or hardwood. It's like heartwood and they call it hardwood, whatever, it's the same. Because it's like more compact xylem, it's not living, it's not like water flowing through it as much. The outer part of the tree, like the superficial part of the wood, that's more active as far as xylem, as far as water flowing through it. I'm not gonna ask you about cork. It's just like, some trees have cork. All right, so what are the things that I'm gonna ask you on the test from this chapter? You know what, I'll stay on this because I don't want to switch back and forth. Number one, the difference between monocots and dicots. Some of the differences. We got 5 different things here, so I don't know, I might say give me 3 differences or tell me all of them, I don't know. But number one, know this slide, know the difference between monocot and dicot. There's one question. Question 2, know the 3 types of plant cells. Giving you the test here. I'm gladly giving you the test here. We all need a win. No fucking turnovers with you guys. Just start studying now. Don't be a hill. Yeah, so types of cells. That's question number 2. 3 types of cells. And remember, thin, unevenly thick, thick. We'll get back to that. Number 3, question number 3. 3 types of plant tissue, vascular, dermal, ground. Give me an example. I guess I'm still on question 3. Give me an example of vascular tissue. You're going to answer xylem, phloem. I know they're 2 words that you got to learn to spell, but xylem and phloem. That's question 3. Question 4. Wow. Okay, now some smaller questions. Maybe I'll have to know what a cuticle is on a leaf. To prevent drying out. It's a waxy layer. Oh, okay. Another question. I want you to know that wood is xylem, and I want you to know that bark is phloem. I would also like you to know, if I ask you what kind of cells are wood and bark, you should be able to say it. So you've got 3 choices, right? Parenchyma, parenchyma, sclerenchyma. You know parenchyma is for new plants. So is wood new? Is it like new growth? It's like, is it getting taller? It's not new growth. Is it the skin? Is wood the skin? No. So xylem is sclerenchyma. Actually, xylem and phloem are both sclerenchyma. I don't know. That's like 5 questions right there. And I'm kind of out of questions here. Yeah. I don't see why you can't get all of those right. Right? That like that part of the test, you're gonna get it. All you gotta do is just it's easy, you just gotta spend time memorizing the words. Right? It's not thought-provoking. I can't say describe roots and stuff like that. There's not any of those types of questions. It's just like, what are the 3 types of cells? And you just answer what you study. So there's no reason to screw this up unless you don't study. So are there any questions on this PowerPoint? This one here. Wow, it's already almost 10. Any questions on this PowerPoint? Let's see if you guys are even with me. That's my fear, like I get on here after. I get on here after like an hour and nobody's left, and it stopped recording, like the internet turned off. I'm still recording. All right, so you guys are here. Well, still recording. Say that. All right, I'm going to open the other PowerPoint, and I'm not going to spend an hour on it. I'm just going to highlight some things that, thank you. I know one of you's here, and I got a student in the class, that's definitely two. So I'll take that. Let me open up the other PowerPoint. And I guess I'll just go over some of the questions because you had a quiz on it, but it was multiple choice. So now I'm going to make it essay. So let me tell you. So I want you to know. And this is the same slides that I used in the video. Okay. If you guys can't see that first slide, let me know. But the title of this slide is Sunbeam, but this is energy. This sunbeam, they're called photons. Not protons, photon, like in Star Trek. P-H-O-T-O-N. I had to think about that. These are photons. There's energy in this light. It's energy, and it comes down in waves. And some of these waves are like super tall, like the wave. They're all invisible. We can't see anything, but it's there. It's energy. We know it's energy because it's heat, right? The sun's hot. The sun gives us heat. Therefore, heat is energy. If something is hot, we know for sure it's got energy. Anyway, some of these waves are really big and some of these waves are very small. Some of these waves carry radio signals, television signals, microwave, whatever, X-ray, all of that. Some of them carry light, visible light. So some of these waves we can see. Most of these waves we cannot see. Some of these waves we can see. And it has energy in it. So they're called photons. I want you to know what photons are. I want you to know the two parts of the chloroplast. So these cells, these are the cells in the leaf, and there's a few things that are true about them. One, if we were to say what kind of cell are they, they're parenchyma. But another thing that we can call them is mesophyll. Yeah, right here. These are mesophyll cells. So they're not only parenchyma, but they're special parenchyma that go through photosynthesis. For example, the part of the potato we eat, those are parenchyma cells too, but they're not mesophyll. There's no photosynthesis happening there. So it's pretty much the cells of leaves. We call them mesophylls. And you can see this mesophyll cell, and it's got a ton of chloroplasts in it. Now I want you to know the two parts of a chloroplast. Well, you have all these green discs. Those green discs, they're all stacked on top of each other. The green disc is called thylakoid. The liquid that it's floating in is called stroma. So I want you to remember that because there's two types of reactions in photosynthesis, light and dark. Or your book might call it this, light dependent and light independent. So light independent is the same thing as dark reactions, meaning that you don't need the light. So light-dependent reactions, we just call them light reactions. Light-independent, we call them dark. So I'm just using the terms light and dark. Light reactions happen in the thylakoid. Dark reactions happen in the stroma. So you need to know that. This is looking inside of a chloroplast, so it's the same thing. Light reactions happen in the thylakoid, dark reactions happen in the stroma. I'm going to skip this one. You know why I put it in there. Here's light, right? And so I want you to know that light objects have pretty much two choices with light waves. They can either absorb it or reflect it. And so the light waves that are being reflected, that's the color that you see. They give numbers here. I don't really care about the numbers so much. These stand for like nanometers, but what it's showing you is that these wavelengths appear as different colors when they're reflected by objects. So if you take a plant, like a typical leaf that has chlorophyll pigment, chlorophyll's reflecting what? Green. That's what you see. Chlorophyll's reflecting green. There's other pigments in plants like carotenoids. They're reflecting orange, yellow, and red. They're absorbing this color down here. You know, they're absorbing like greens. They're reflecting oranges and yellows and reds. So right now in like New England, the leaves are changing color. Why? Because they stopped making chlorophyll. But they're still making carotenoids. So you can see the carotenoid pigment, but you cannot see the chlorophyll anymore because they stopped making it. The plant, the leaves are going to die, right? So before they die, they stop making the chlorophyll. Now you don't see the green anymore. Now you see the other pigment is like underneath. Then after a couple of weeks, like up in Maine right now, those trees are they're stopping making the carotenoids, right? And now the leaf is going to just die completely. But what you see reflected is the color you see. So that was one of the questions I put on the quiz. I forgot what colors it was. Maybe it was like red and white or something like that. You know, whatever you see, that's what's being reflected and not absorbed. And so what happens is that when you absorb the photons, when you absorb the light, it excites the atoms. It excites the hydrogen atoms, and the electron jumps out of its orbit, and the electron's caught. So that's energy. When you catch electrons, that's capturing energy. I mean, think about what we do. We get electrons from our food. If you think back to that whole Krebs cycle crap that's over now, that was what the Krebs cycle was about, right? Harvesting electrons. We're taking the electrons off of our food. Plants are taking electrons essentially off of water. And they're using the sunlight to do it. The sunlight gets those electrons all hyped up, and those electrons are bouncing out of their orbits. And the plant captures them. Same thing happens in, like, I don't know, like in your car, right? If you touch the hood of your car in the middle of summer, it's hot. Well, why is it hot? The sunlight, the photons are hitting the paint in your car, and they're getting those electrons all excited. And the electrons are jumping out of their orbit. But then the electrons return right back to their orbit. And then heat is given off, like heat goes out of the car. Sometimes, like, you know, when you're driving along a long road and you see like that wavy, you can see it's kind of like wavy when you look far ahead down the road. That's what you're seeing. You're seeing the heat come off of the road. Because the sunlight is hitting it, it's making the electrons get all excited, but they're not getting caught. The electrons aren't getting caught. They're just getting excited and heat is given off. Plants are different. If you touch a plant on a hot day, the leaves aren't hot because the electrons are getting caught and the plant is going to use it. I'm going to go to the different slide and then I'll come back to this. Remember, this is the light reaction. In the light reactions, we have two parts to the light reaction. There's photosystem 2 and there's photosystem 1. And I know that's confusing. It's just the order that they discovered it. They discovered photosystem 1, and then later, scientists discovered that there's another photosystem that's actually before it. So they just called it photosystem 2. It's kind of confusing. If you look at this slide, there's a different name for it. Photosystem 2 is also called P680. Photosystem 1 is called P700. That's like another name for it. But anyway, let's look at what happens. So these photosystems are just like groups of pigments, groups of chlorophyll pigments. In photosystem 2, the photon is getting knocked out of its orbit. And this is kind of a good analogy. So it gets, you see the photons like the hammer and it's knocking the electron out of its orbit. Normally what happens is that this electron comes up and then returns right back down. Normally in things that are not plants or solar panels, things that are not plants, the electron gets knocked out of its orbit and then returns right back again. But here in the plant, it's different. The photon is knocking the electron out of its orbit and it's getting caught. So now we can take that electron and use it to make ATP. So photosystem 2 is going to send this electron down the electron transport chain and make ATP. So what do we get out of photosystem 2? We get ATP. That's the whole point of light reactions. We want to get some energy saved up. We want to make ATP, and we want electrons. They're both energy. It's like a Prius, right? The Prius doesn't have just gas in it. It's also got a battery. It's both energy. The gas is going to make the car go, and so will the battery. And the Prius switches off between battery and gasoline, depending on what you're doing. Plants are the same, except here it's electrons and ATP. The plant is using both. So we need both sources of energy. It's not like us humans. With us humans, we want ATP. That's it. Plants, no, they can use both. They use ATP and electrons. So plants are like hybrids. So photosystem 2 is going to just make ATP. That's it. Now follow the electron down into photosystem 1. Photosystem 1, same thing. The sunlight is hitting photosystem 1 and knocking these electrons out of their orbit. But now, he's going to stick it in this bucket. We're going to save the electron. Remember, just like in cellular respiration, you have these molecules, NAD, FAD. Here they're calling it, you see the bucket, they named the bucket NADPH. And we never talked about oxidation and reduction. So I just told you guys, NAD is an electron carrier, FAD is an electron carrier. It doesn't matter what form it's in, NADPH, NADP plus, whatever. NAD, electron carrier. So we got photosystem 2,
It's just making ATP. You have photosystem 1, it's just storing electrons. So that's the difference between photosystem 2 and photosystem 1. Photosystem 2 makes ATP. Photosystem 1, get the electrons. This is both, so I want you to know the difference between photosystem 2 and photosystem 1. The difference is whether it's for ATP or it's for electrons. And this is both energy. These are both light reactions. This is both light reactions. So this is happening in the thylakoid. So remember, light reactions happen in the thylakoid. And you need sunlight for it, right? This was not happening in the nighttime. The sun's down. None of this photosystem 1, photosystem 2, it's nothing. Nothing's going on. There's no photons getting knocked out, whatever. So it's light reactions. The purpose of the light reactions is to take the energy from the sun and convert that into electrons and ATP. Once we have a lot of electrons and ATP, we can take it and make sugar with it. So here's another question. We keep knocking electrons out of their orbit and catching them, which means they never go back. You can't keep doing that. You're going to run out of electrons, right? You can't keep knocking them out of their orbit and they don't ever come back. So where do we get the new electrons? We get them from water. So how do we replace the electrons that have been taken? Water. The plant knows how to take H2O and it splits it. It takes the H's from the H2O from water, it takes the hydrogens, and it pulls the electrons off of there. What happens to the oxygen part of the water? It just goes back into the air. The plant is making oxygen for us. It's taking water, giving us the oxygen, putting the oxygen into the air, and it's using the hydrogen, it's taking the electrons off of the hydrogen. So I want you to know where plants get their electrons. They're splitting water, that's the answer. They're splitting water. And that takes energy, right? I mean, where are you going to get the energy to split water? Well, from the sun, sunlight. So this slide is probably like the easier slide, so I'd like to just stay on that slide. This is just showing you that when we talked about the electron transport chain in animals like humans, we were talking about the mitochondria. And if you remember, like, all the H pluses go to one side and then they all go through and then it puts the ADP and the P together. I don't know if you remember all the H pluses going. That was the electron transport chain. Plants do the same thing. They just do it in the chloroplast. But it's like the same process. Am I going to ask you about that? No. But it's just for your information. So it's the same process. All right, here we go, Calvin cycle. Obviously, I'm not going to make you explain the whole Calvin cycle. But there are some things that I want you to know from this. One, you're joining, well, the first thing I want you to know is how do we make sugar? How does the plant make sugar? It's making it from carbon dioxide. It's making it from carbon dioxide and water. So if you think about sugar, there's 3 atoms that are in sugar. There's like 3 ingredients to make any kind of sugar. Carbon, hydrogen, oxygen. Well, the hydrogen and the oxygen, where do we get that? You get that from water, right? Because what is water? Water is H2O. Is there any carbon in water? No. We need carbon. So we got the hydrogen, we have the oxygen, we need carbon. Then we can make sugar. Where do we get the carbon? Carbon dioxide from the air. So that's what the Calvin cycle is doing. This is the dark reaction. This happens in the stroma. This happens in the stroma of the chloroplast. So we have to get the carbon dioxide out of the air. The plant is going to take this carbon dioxide and it's going to join this to a molecule called, and you could just know the abbreviation, RUBP. And I want you to know this because I've seen this on like entrance exams, like the T's or the HESI. I've seen this question. There's two molecules that I see them ask. One is RUBP, the other one's rubisco. So RUBP is the molecule that we're going to join the carbon dioxide to. This looks like a Krebs cycle, but it's different. The Krebs cycle was catabolic. The Krebs cycle was all about breaking up something, right? It tore up that acetyl-CoA. This is the opposite. This is making. This is building something. This is analog. We're making sugar. So this is going around and around and around. The RUBP is coming around and grabbing the carbon. And every time and rubisco is the name of the enzyme that is going to join these two molecules. So rubisco is an enzyme and it joins carbon dioxide. So it's joining the carbon dioxide to the RUBP. So I want you to know what rubisco is. Because there's probably like 4 billion tons of this if you were to weigh it all on the planet. It's like the most common enzyme on the planet. So we're not going to explain the whole thing. It actually gets a little bit complicated because you have to run through this process a lot of times. But just look at it this way. Every time this thing goes around 3 times, it's picking up 3 carbon dioxides. So if you look at the input, 3 carbon dioxides. If you look at the output, every time it goes 3 times, you end up with a half of a glucose molecule, which we're calling G3P. You might remember that from the last chapter, the chapter on cellular respiration. So think of G3P as like half of a glucose. So if this were to go around 6 times, you can make a glucose molecule. So for a plant, G3P, you could take glycerol di3-phosphate and you could make it into different types of sugars. Cellulose is a type of sugar a plant can make. That's the cell wall. Starch is another type of sugar. I shouldn't use the word sugar. I should say carbohydrate. You could take G3P and make it into different types of carbohydrates. Starch, like a potato. Cellulose, like plant walls. Or you could just make it into glucose. So that's why they put G3P here. So anyway, you need to know that the Calvin cycle is the dark reaction. Calvin cycle happens in the stroma, because that's where dark reactions happen. And it's all about putting together RUBP with the carbon dioxide. Shit, I already drank too much coffee today. All right, here's the last thing I want you to know. The last thing I'm going to test you on. So look up here. You see how ATP is being, there's like, it's up here twice. But then NAD is up here only once. Remember I told you the light reactions get two types of energy, ATP and electrons. Well, here the plant is using ATP more than the electrons. So the light reactions, we get ATP and electrons, but then in the dark reactions, we're using more ATP. So what's going to happen after a while? You're going to have excess electrons. So now the plant has a problem. We got like too many electrons, but we don't have enough ATP. We keep getting extra electrons. So now you got to go back to the light reactions, and we need to just make ATP. So you got to like balance it back out again. And so this is the part that's on the test. This is called cyclic, CYCLE, cyclic electron flow. So here's what's going to happen. Check out this dude up here with the t-shirt, not the tank top. He's going to take that electron, he's going to throw it back down onto the ramp. He's not going to stick the electron in the bucket. He's just going to put it right back onto the ramp. Let's follow this electron. It's going down the ramp. It's making ATP. It gets knocked back up to him again. He catches it again, throws it back down the ramp. See how it's going like in that circle, the cycle? So it's like an electron flowing in a circle. Cyclic electron flow. We're just making ATP. He's not dropping it in the bucket. He's just throwing it back down the ramp again. So that's what will happen when you want to make more ATP to make up for the deficit. We call it cyclic electron flow. As you see, it doesn't involve photosystem 2. She's not doing anything. She's just holding on. He keeps throwing it down here. He keeps throwing this electron back into the electron transport chain. So it only involves photosystem 1, not photosystem 2. And that's just to make ATP. Because we keep using more ATP.
It only uses photosystem 1, you said? It was
Correct.
photosystem 2 because it's making ATP.
And remember, photosystem 1, oh yeah, I get what you're saying. Photosystem 1 is only for making ATP. I'm sorry, photosystem 2 is just for making ATP. Photosystem 1 normally is just for storing the electrons. So normally this guy's job is to what? Put the electron into the bucket. But sometimes there's like a special case where you only need to make more ATP. We're not going to have her, because if she sends this electron down the ramp, it's just going to go up to him anyway.
Okay. So cyclic electron flow, it's only using photosystem 1.
Yeah, cyclic, he's actually making the electrons. She stops. He's telling her, hold on. He's going to make it. He's going to throw it back down the ramp. So normally, if I were to ask you what's the difference between photosystem 2 and photosystem 1, we're talking about like a normal case, then you're going to say photosystem 2 is for making ATP. Photosystem 1 is for just collecting the electrons. But then there's like a separate question, what happens during cyclic electron flow? Now it's a little different. Photosystem 2 is going to throw the electrons back down onto the ramp. Eventually, he's going to make enough ATP, and then he's going to just start putting the electrons in the bucket again. I know, I get what you're saying. That's like a little confusing. So only in the special cases where you end up with too much ATP, I'm sorry, too much electrons and not enough ATP. Now you're going to switch. You guys have any other questions? That's my last slide. So I'm going to ask you, what is cyclic electron flow? You need to know the difference between light and dark, like where do light reactions happen, where do dark reactions happen, what part of the plant. What's the point of the dark reactions? The point is to take the electrons and the ATP and make sugar out of it, glucose, or you could just say sugar. What's the point of the light reactions? Take the sunlight and make the ATP and electrons. Within the light reactions, there's two types, right? Photosystem 2 and photosystem 1. So you should know the difference between them. In the Calvin cycle, or the dark reactions, I want you to know what, that RUBP is being joined up with carbon dioxide. What's the enzyme that's doing it? Rubisco. That's pretty much, that's everything. I mean, that's all my questions. I'll be lucky if I get 10 questions out of this. But you know what they are. All you got to do is just, between now and Wednesday, just study. Come in and take the test, just like last Wednesday. Same room, same schedule, same everything. It's going to take you, you're not going to need 45 minutes. No one used 45 minutes last time. You guys were doing it in about 20 or less. All right, so I'm going to stop this recording. It's probably too long as it is.